In this lecture, we're going to be spending a lot of time discussing brain areas and their functions. The important thing to keep in mind is that one brain area does not work on its own to perform a function. So you'll notice that there'll be several brain areas, depending on what level of the brain we're in, that have similar functions. And it's likely that those brain areas work together to perform those functions. But for the sake of this course, we're going to assign specific brain functions to brain areas, but I want you to keep in mind that no brain area works in isolation to perform a function. Let's get started. Before we dive into brain areas and their functions, we need to talk about what your textbook calls the brain's geography. But really, we're using terms to orient ourselves once we're in the brain. And these are terms that neuroscientists as well as physicians use to orient themselves to different areas of the body. We're going to be using it largely for the brain, but you can apply it to any body part. So let's start over here with this. Actually, let's start with the cat, because I love kitties. They're one of my favorites. So you want to imagine kind of like a line going through the middle of the cat. That's the midline. The midline here is kind of through the middle, dividing this person into two symmetrical halves. But let's talk about A first. When we're talking about the rostral portion or something that's rostral, we're talking about structures that are closer to the nose, because the term rostral literally means towards the nose. The opposite of rostral is the term caudal, and that means towards the tail. Okay, so structures that are rostral are going to be closer to the front. Structures that are caudal are going to be closer to the back. The term dorsal means towards the back. So we're talking about the back surface of a person, the brain, a cat. Ventral represents areas closer towards the belly, right? You can see that. This is the belly. And so when we're talking about human beings, because we don't walk on all fours, and so our nervous system actually, the midline bends, unlike that of a cat. I don't know why these lines keep showing up, but they do. I'm not drawing them. So when we talk about rostral, this is the bend that we're referring to. So what changes is the term dorsal. The term dorsal means top of the brain, but it also means towards the back once you get past that bend in the brain. Ventral is the same thing. Ventral means towards the surface of like your chin area, but it can also mean towards the belly once you get down past this bend. Okay, so just keep that in mind when we're talking about dorsal and ventral in the human brain. Because of this bend in our nervous systems, we have to keep in mind what we mean by dorsal depending on where in the brain we are. Superior means on top of, and the opposite of superior is inferior. Anterior and rostral are relatively the same. Anterior means towards the front. Posterior is the opposite of anterior, that's towards the back. Okay, so these are some of the terms that we're going to use when we're discussing brain orientation. The other couple of terms that I want to refer to are medial and lateral. Again, everything is relative towards this midline, this imaginary midline. And so medial means these structures are closer to the middle of the brain, and lateral means the structures are closer to the outside of the brain. So for example, your ears are lateral to your nose, and conversely, your nose is medial relative to your ears. Okay, and then when we're looking at superior, this orients you to the top. Inferior would be towards the bottom. The last couple of terms that, the orientation terms that we haven't talked about, are proximal and distal. These are typically used when we're referring to appendages, so they're rarely used in neuroscience because I mean, unless we're talking about nerves innervating or connecting to distal or proximal musculature, we rarely talk about them in terms of orientation of brain structures. But just so you're aware, proximal, again, proximal and distal refer to the trunk. Just like everything prior is relative to this midline, proximal and distal are relative to the trunk of the body. So when something is proximal, it's closer to the trunk of the body. When something is distal, it's further away from the trunk of the body. So your fingers are distal to your shoulders are proximal to your elbow, right? So I think you just have to keep that in mind. All of these terms are relative to a central midline or the trunk of the body. All right. The very last thing we're going to talk about are planes of view of the brain. I got to erase this because the lines are kind of getting in the way. Oops, back to the pen. The last thing we're going to do is talk about the planes of the brain. We can view the brain in 3 different planes. The first one we're going to talk about is the coronal plane. And again, it's just showing you one slice, but you can slice the brain through this plane. So the coronal plane, all of these are kind of flat panes, kind of glass that you can imagine. And they're firm, they're not wiggly, so they're straight lines. And so the coronal plane allows you to kind of slice through the brain, to use the terms that we just covered, rostrally to caudally, right? So we can have slices going this way, right? Oh, I forgot to orient you guys. My apologies. I assume you know what you're looking at here. But just so you're aware, the nose of the person would be here, the back of the head is here, and the top of the head is here, right? So it's similar to this brain orientation shown right here. So the coronal plane, we're slicing the brain through sections from rostral to caudal. The horizontal plane, we're slicing the brain dorsal to ventral. Okay. And I'm going to erase this because it's getting hard to see, but the sagittal plane is a little bit unique because we're going, I'm going to use this head here because it's easier to see. In the sagittal plane, we're going from outside to inside or from inside to outside, right? So to use the terms that we just learned about, sagittal plane, we're cutting slices medial to lateral or lateral to medial. That's the view of the brain that we're getting. Okay. All right, let's continue. Now we are going into actual brain structures. So it's easier for us to see brain regions based on kind of groups. And there are 3 major divisions of the brain based on its development. So initially, in early stages of development, the brain only has 3 major parts. They are forebrain, midbrain, and hindbrain. And just to give you an idea, in case you're unfamiliar with it, fore means the front, hind means the back, and mid is sandwiched in the middle. Okay, so the forebrain would be closer to your head, hindbrain would be closer to your spinal cord, and midbrain is sandwiched in between. As the brain develops and gets more complex, the neuroscientists or neuroanatomists added a subdivision related to each of these 3 major divisions because those subdivisions are, I guess, related to each other in terms of their development. So from rostral to caudal, or anterior to posterior, we are talking about the telencephalon. It's the topmost area of, or the frontmost area of the brain. Then we have the diencephalon, mesencephalon, metencephalon, and myelencephalon. And these are a lot of cephalons. The worst cephalon means head or brain. But the way you can remember the order from anterior to posterior is T is top, and then the rest of them fall into alphabetical order. Okay, we're going to be focusing on this last column here. So I would recommend that you create a chart of your own with these principal structures. And if you want to include the major division and subdivision, you can. But then you want to have a column to the right of the principal structures that focuses their function. This focuses on their functions. This will give you a nice kind of chart to reference when you're looking at kind of structures of the brain and what they do, and learning all of them. It'll be in one single page as opposed to over multiple slides that we'll be covering in this lecture. All right, so we are going to be talking about the principal structures within each of these subdivisions, starting with the telencephalon and then moving our way all the way to the myelencephalon. So in the forebrain, we have the telencephalon and the diencephalon. We're going to focus on the telencephalon first, the topmost or anterior or rostralmost structure in the brain. The telencephalon includes the cerebral cortex, the basal ganglia, and the limbic system. And we'll talk more about these structures in 1 second. So generally speaking, the cerebral cortex is that large convoluted structure that you see on the outside of the brain when you're looking at the human brain. Both the basal ganglia and limbic system are not easily visible unless we cut through and dissect into the brain, right? So the outermost surface of the brain is the cerebral cortex, and you can't really see the other two structures of the telencephalon, the basal ganglia and the limbic system. The cerebral cortex is assigned to have higher order functions that we'll talk about later. So if you want to think of like a person in charge of the brain, that would be the cerebral cortex. The basal ganglia handles largely voluntary motor function. The limbic system is called the emotional centers of the brain because it handles structures that process emotion, but it also has structures that handle memory, because a lot of the time emotionally charged things are linked to memories in our mind, and it also handles motivated behaviors, because oftentimes things that we're willing to do or want to do are driven by our emotions. So it's considered to be part of the limbic system. Let's talk about the cerebral cortex first. So the cerebral cortex is the outermost surface of the brain. It's what you can see. The word cortex means bark. And you'll notice, or from the pictures, or if you've ever seen, videos of the human brain, it has lots of convolutions, like kind of bumps and ridges and grooves. These convolutions, as we went through evolution, helped increase surface area without making our heads giant. So by folding in on itself, the brain created more surface area to house more neurons to allow us to engage in more complex behaviors. The convolutions have scientific names. There are grooves and bulges. There are shallow grooves or smaller grooves called sulci or sulcus. The larger grooves are called fissures or fissure for singular. The bulges, all of them, there's no big bulge or little bulge, all the bulges are known as gyri, gyrus for singular. The cerebral cortex is made up of two identical hemispheres, so there's a giant fissure that kind of splits the cortex in half, and that's called the longitudinal fissure. And what bridges the two hemispheres is a white matter axon-filled structure called the corpus callosum. There are other white matter tracts. So when I refer to white matter tracts, I'm referring to bundles of axons that make up structures within the brain, and these white matter tracts contain largely axons, and they carry information from different areas of the brain. But there's so many of them bundled together that they appear white. So that's why we call them white matter tracts, because they're covered in myelin. Internal capsule is a white matter tract that links the cortex with the thalamus. When we talk about the thalamus, you'll know why we have so many axons connecting the cortex and thalamus in particular. The anterior commissure connects the hemispheres, but it does so kind of on the bottom half of the brain, ventrally. So the corpus callosum can be found in the dorsal surface of the cortex, whereas the anterior commissure, you're going to find them connecting things closer to the base of the brain, or ventral surface of the brain. When we take a look at the cortex, we can divide them into 4 lobes, and these lobes were given their names based on the bones of the skull under which they reside. We have frontal, parietal, temporal, and occipital. And functions have been assigned to these lobes, but it's important to keep in mind that as we get more and more complex, and as there's more and more overlap, because again, our brain does not work in like silo functions, rather, it coordinates function across multiple actions, that there's bleed through in their functions near the borders of these lobes. All right, let's take a look at the 4 lobes and what they do. So just so you're aware, this line here divides the frontal from the parietal. So this is the parietal. Oh my God, I don't know why those lines keep popping up. This is the occipital here, and this is the temporal here. Okay, so forgive the lines. I can't seem to control that. PowerPoint just keeps adding it in. All right, so let's take a look at the frontal. I'm going to erase these lines just so you can see the lobes better. So the division of, actually maybe the laser pointer is better. The division of the frontal and parietal lobe is right here, right? So the front half is the frontal, the back half is the parietal, and then we have this arbitrary line kind of dividing the occipital lobe from both the parietal and temporal. And then we have the temporal lobe kind of sitting on its own. It's got its own massive fissure, the Sylvian fissure, breaking it up. Okay, so back to here. The frontal lobe is in the frontmost area of the brain. It's responsible for higher order functions like reasoning, decision making. And because the primary motor cortex lives in the frontal lobe, it also handles movement. Okay? The parietal lobe handles sensory processing. So it's getting information from the skins, body position from the muscles, as well as internal organs. So anything involving sensation, like touch, is going to be handled by the parietal lobe. And we have what's called the somatosensory cortex living in the parietal lobe. Soma, as you know, is another term for the cell body. So this term, somatosensory, means sensations emanating from the body. And that's what the parietal lobe handles. The occipital lobe has only one function, and that's vision. And it's because human beings rely so much on their sense of vision that we've devoted a whole cortical lobe solely for vision. The temporal lobe, which you can likely not feel, but it's positioned right behind your temples. That's why it's called temporal lobe. It's responsible for hearing, language, emotion, and memory. The structures responsible for emotion and memory actually live deep within the cortex, so you can't actually see those structures, but they are part of the temporal lobe. Okay? So again, keep in mind that these functions that have been ascribed to these lobes are not all-encompassing. Here I'll give you an example. There are parts of the visual cortex that coordinate with the parietal lobe to handle hand-eye coordination, right? And so you'll find as we get higher up into the processing of any particular sensory system, you're gonna find coordination between the lobes to handle those functions. Okay, so we've already talked about these specializations of the different lobes in the prior image, but I want to highlight or add to that image, and it's the prefrontal cortex. So let me go back to that image real quick. The prefrontal cortex is right here. It's the frontmost area of the frontal lobe. It is the structure that is the most recently developed with regard to evolution, and it handles what we call executive functioning. So these are functions that make us uniquely human. things like planning days, months, if not years in advance, right? Saving money so that we can purchase something bigger rather than spending it on something that we want. Delayed gratification. Decision making. It also handles working memory, which is holding information in our mind until we're ready to use it. It is the seat of our personality and behavioral inhibition, kind of squashing this urge to react even though we might want to, right? We might have the urge to react and act, but we know that it might not be socially appropriate, and so we inhibit that reaction, and that's what the prefrontal cortex does. In addition to being the most recently evolved structure, it is also the very last structure to develop. The prefrontal cortex is not fully developed until you're 25 or so, thereabouts. And so the brain is not fully developed. The prefrontal cortex, which handles things like, understanding consequences of your actions or delaying gratification, that is not fully developed in the teenage brain or even in your early 20s. And oftentimes people make decisions that aren't fully well formed or thought out, and it's because their prefrontal cortex is not fully developed. The cortex is very, very thick. It's, relatively speaking, compared to other brain areas, it is made up of 6 different layers of types of cells. The biggest cell type, and the one that's the most beautiful, is the pyramidal cell layer. And the reason why it's called pyramidal cells is because these cells have, their cell bodies are shaped like pyramids. So there's an inner layer and an outer pyramidal cell layer. These cells perform different functions based on the input that's coming to them. The pyramidal cells are the ones that are responsible for kind of sending information out of the cortex. But you don't need to really worry about what these different layers do, but just understand that there are 6 different layers. They develop differently, inward to outward, and they also house different types of cells that perform different functions. The next structure in the telencephalon is the basal ganglia. This basal ganglia is a collection of structures. It's not one area. I didn't know that when I first learned it, but it's really a bunch of different structures that have been grouped together because they work together to control voluntary movement. They include the caudate nucleus, the putamen, and the globus pallidus. So these 3 structures together make up the basal ganglia. Sometimes, especially in lower order animal models like rats and mice, they don't have a separate caudate nucleus and putamen. So they sometimes, the combined caudate and putamen is known as the striatum. So there's no really clear delineation of the caudate and putamen in rats and mice's brain. And so you might often see the word striatum, but it's referring to the caudate and putamen collectively. If you know or have heard of Parkinson's disease or know anybody who has Parkinson's disease, you might be curious about this piece of information. In Parkinson's disease, there's an area of the brain that resides in the midbrain that we'll talk about later called the substantia nigra that projects, so it sends axons to the basal ganglia and releases neurochemicals to it to activate it. And in Parkinson's disease, the substantia nigra neurons die, making it harder for them to perform in a voluntary movement. And that's because the basal ganglia has to receive input from the substantia nigra to be activated, to activate voluntary movement. And as the substantia nigra dies, the person with Parkinson's disease finds it harder and harder to perform voluntary movements. So here's the basal ganglia. You really can't see it from the outside, so they've created kind of like an image of the basal ganglia within the brain, if you could see it. So there are 2 kind of basal ganglia located, one in each hemisphere, and you can kind of see roughly where it is. The thalamus is located on the innermost medial-most area of each hemisphere, and just outside of the thalamus, we've got the globus pallidus. Okay. All right, on to the limbic system, which is the last kind of category of structures within the telencephalon. The limbic system are structures that kind of surround or circle the thalamus. They are largely handling emotion-focused functions. So they handle motivated behaviors, emotion processing, typically negative emotion, as well as learning and memory. And that's part of the reason why it's a lot easier to learn something if you have a strong emotion tied to it. The structures that are in the limbic system that are important for our course are the hippocampus, the amygdala, the cingulate cortex, and the septum. You may have heard of the hippocampus and amygdala before if you've taken enteropsych, for example. The hippocampus handles learning and memory. The amygdala processes negative emotions such as anger and aggression. And so here in kind of green and a little bit of red and purple, you have the limbic system. So the thalamus is here, this kind of egg-shaped structure here in the middle, and you'll see the limbic system involves structures all that surround the thalamus. So we've got the cingulate cortex in green here, the septum is in blue, the hippocampus is in purple, and then the amygdala is in red. Okay, and you'll notice that there are some connections between these structures so that they can coordinate their efforts with regard to emotional functions. All right, within the forebrain, in addition to the telencephalon, we have the diencephalon. This is a little bit easier because they're simplified structures and there's only two of them. We've got the thalamus and the hypothalamus. Hypo means below. So then, not surprisingly, the hypothalamus can be found below or ventral to the thalamus. But both of these structures make up the diencephalon. The thalamus serves as the sensory relay center of the brain. What that means is no sensory information gets to the cortex without first going through the thalamus for processing first. The hypothalamus, which we'll also expand on, serves as the endocrine and autonomic control center. So endocrine systems involves controlling hormone release from glands all over your body. And the master coordinator of that release is the hypothalamus. So it coordinates and decides when and how much hormones get released from different glands all over your body. It also handles the activation of the autonomic nervous system, or parts of the autonomic nervous system, like the parasympathetic division. So let's take a look at where we are in the brain. So here's the one half of the hemisphere. So if we were to cut the brain directly down the midline, we would get a symmetrical half. This is the right half of the brain. And so the nose would be here, the back of the head is here, this is the top of the head, and here is the diencephalon. If we blow it up and bring it here, here is the thalamus, right? And then here, peeking out through, because you can't really see it, is the hypothalamus. So the thalamus is much, much larger than the hypothalamus, and the hypothalamus is located in the middle ventralmost area of the diencephalon. So it's like right here. So this is the hypothalamus and the rest is the thalamus, and it extends outwards laterally. To give you an idea of what I mean by the thalamus being the relay center, all sensory information from eyes, ears, skin hits the thalamus first before it gets sent to the cortex for processing and conscious awareness. So the thalamus is getting a crap ton of sensory information that may or may not get sent to the cortex. But remember early on when I talked about the white matter tracks, one of them was the internal capsule. The internal capsule exists solely so that structures in the thalamus that process sensory information can send their information to the cortex. That's how connected these structures are. And when we talk about the motor cortex communicates with the thalamus then to send that information to the muscles of the body. So the thalamus and cortex have a very strong relationship, and the internal capsule connects them both. All right, we're ready to move on now to the midbrain. We're done with the telencephalon, and we're entering mesencephalon and midbrain. And so what you're looking at here is the back half of the head. So here's the thalamus, right? This is kind of like where the back of your head would be, and this is where your neck would start. And they've taken away the cortex completely. So you can actually see some of the structures that live in the midbrain. We're going to be talking about some of these structures that are outlined here. Don't worry about the 20s. We're going to be focusing on 1, 2, and 3, as well as some other structures that are deep within the midbrain that you can't see. So this is an actual human brain dissection, so not just a cartoon. All right. So the mesencephalon, or the midbrain, is also known as the mesencephalon, and there are two collections of structures in the mesencephalon, the tectum and the tegmentum. The tectum, which means roof, because it's on the top half, consists of two structures, which you can actually see in the prior picture I'll show you in 1 second. The word colliculus means little hills. There's a superior colliculus and an inferior colliculus. So let me go back. So these are the little hills. So this is the superior colliculus, one on either side. And here's the inferior colliculus, one on either side. Okay, superior is on top, inferior is below. Okay, the superior colliculus handles visual reflexes. So what do I mean by visual reflexes? If you're looking at something, like you're watching TV, and something moves in the corner of your eye, and you shift your vision to it, you can thank the superior colliculus for that, because that's what allowed you to kind of shift your view quickly without even thinking about it to whatever was catching your attention in your peripheral vision. The inferior colliculus does pretty much the same thing, but it does it for the auditory or hearing reflexes. So the fancy word for hearing is audition. And so the inferior colliculus diverts our attention. If we hear a loud sound in the room that we're in, we're going to turn our heads towards that loud sound to try to identify its source. Okay, so that's the tectum. That's within the midbrain or mesencephalon. The bottom half of the mesencephalon is made up of the tegmentum. It consists of 3 colorful structures and the VTA, the ventral tegmental area. So the 3 colorful structures are periaqueductal gray, substantia nigra, black, and the red nucleus. The periaqueductal gray is really important for pain processing and pain reduction. If you've ever heard of endorphins and opiates act in the periaqueductal gray to reduce pain perception or pain experience. The substantia nigra, which we've already talked about earlier in this particular lecture, sends its axons to the basal ganglia. So it handles motor function, and it's the one that dies in Parkinson's disease, PD. The red nucleus also handles motor function, but different kinds of motor functions. Again, you don't need to worry about it because we're not going to be going into depth with the motor system at all in this course. And then the most important that we'll be going into greater detail in this course is the ventral tegmental area, or VTA. The VTA is part of the reward pathway or reward system. Activation of the VTA typically is associated with things that bring us pleasure or that we enjoy. And so it's not surprising then that this particular structure is also activated in the disease of addiction. And we'll talk about addiction later on in the semester. So to kind of orient you to where these structures are, so this was the hypothalamus, This is the thalamus. Here we have the midbrain. Whoa, that went just crazy. Hold on, let me just use the highlighter, not the highlighter, the laser pointer instead because my pen isn't working. So here is the midbrain. We have the superior colliculus, the big hill, the upper hill, the lower hill, inferior colliculus. The tectum is what the superior colliculus is made of. And then you have a space in between that kind of separates the tectum from the tegmentum. The tegmentum sits below and consists of, and here's a slice of the tegmentum. We have the periaqueductal gray, right? It's called periaqueductal gray because it surrounds this space called the aqueduct, and it's gray because it's filled with cell bodies of neurons. We have the red nucleus that has slightly reddish color to it when sliced in dissections. And then we have the substantia nigra, which is actually pigmented black. The word nigra is another word for black. The substantia nigra is pigmented black because of the byproduct of making some of the neurochemicals that it makes. So collectively, this is the tegmentum, and this is the tectum, the top half. But this whole thing is a slice through the midbrain. Oh, apparently I can't advance with this, so switch back to my pen. Now we're going to move on to the metencephalon and myelencephalon, which make up the hindbrain. So we've talked about the forebrain and midbrain, and now we're transitioning into the hindbrain. So the metencephalon, which is anterior or rostral to the myelencephalon, is made up of the pons and the cerebellum. So the pons is visible, if we kind of looked at the bottom surface of the brain, is visible easily, as is the cerebellum. But in purple here, that's your metencephalon. And then the myelencephalon has just largely one structure, and that's called the medulla, and it's shown here in light blue. So let's talk about the two different structures. So here's pons, and here's the cerebellum. Collectively, they both make up the metencephalon, which we're going to talk about next, and it's the first structure in the hindbrain. The pons is the major bulge on the ventral surface of the brain stem. So the midbrain and hindbrain collectively make up the brain stem. In the pons, when you slice through it, you'll see lots of white network-like structures. That's because it's got lots of fibers that are going up and down through it to relay signals from the medulla to the brain. The pons is serving as like kind of this massive switchboard. It also has a crap ton of connections with the cerebellum. So the pons gets a lot of sensory information from the spinal cord that are bringing that information in through the nerve fibers, and it sends it to the cerebellum, which allows us to modify our movements in real time. So the cerebellum handles sensory motor functions. It makes adjustments to our movements based on the sensory information that we're getting, like body position, vision, hearing, all of that. The coordination of all of that is handled by the cerebellum, and it's getting that information from the pons and then sending signals out through the spinal cord to change or modify our skeletal muscles as it sees fit. So it receives a lot of input from the spinal cord and pons to calculate the sequences of movements of muscle contractions to, in target, achieve our movement goals. From the spinal cord, it's getting information like body position, and from the pons, it's getting information from the cerebral cortex. So really, the cerebellum serves as a real-time modification of motor functions. And just to give you an idea of what the medulla does, it is the location where information crosses from one side to the other. So one of the things I haven't mentioned but is really, really important is that all motor function and sensory information from one side of the body is handled by the opposite side of the brain. So information from our right half is controlled by our left hemisphere, and information from our left half is sent to or controlled by the right hemisphere. The point at which this crossing occurs is in the medulla, at what is called the pyramidal decussation. So all the information crosses from one side of the brain to the opposite side of the body at the pyramidal decussation of the medulla. And so here's the medulla. We're ready to talk about it. In addition to carrying those fiber tracts that are crossing from between the brain and body, the medulla, being the most posterior division of the brain and the myelencephalon, handles what are called vital functions. It handles respiration, sleep, attention, cardiovascular function. So if you were to damage your medulla, you would need something like the machine to help you breathe because it can no longer do that for you. Okay, the same thing with cardiovascular function. You would need a machine to help keep you alive if you were to damage your medulla. And so just to give you an idea of the hindbrain, we have the pons and the cerebellum, which make the metencephalon, and then we have the medulla, which makes up the myelencephalon. Collectively with the midbrain, these structures make up the brainstem, and so everything that's not the telencephalon, or in the telencephalon and diencephalon, is considered to be part of the brainstem. And so we started off with this chart, and hopefully you were able to create another column here so you could write down information related to these structures and their details. But that is it. We don't have a summary of these of this information because it's all contained hopefully within your chart, or you will make one in your chart since this information is much easier to grasp in a table than it is in words. Since it's much easier to grasp this information in a table rather than in sentences.